everyone and welcome to Miss Estruch Biology, and in this video I'm covering everything you need to know for topic 6 nucleic acids and protein synthesis for Cambridge International A-level. So you've got all the information you need in one place. Grab a pen and paper to make notes, or if you want to skip that step, then I've got notes ready made for you highlighting all of the key marking points and with examiners' special tips. But for now, let's get into the information. So here is topic 6, and in this topic we're going to be looking at nucleic acids and protein synthesis, starting with 6.1, which is the structure of nucleic acids and replication of DNA. DNA and RNA both occur in the monomer form and as the polymer form, and the monomers of DNA and RNA are known as nucleotides. And the nucleotide consists of a nitrogenous base, which for DNA can be either cytosine, thymine, adenine or guanine, and for RNA it could be uracil instead of thymine, cytosine, adenine and guanine. Both of them then have a pentose sugar, and for DNA that is deoxyribose, and for RNA it's ribose. That's why there's the D and the R difference. And then they both have one phosphate group. Now with these bases, the nitrogenous bases, you can categorise them according to their structure. Adenine and guanine are classified as purines, and that means that the nitrogenous base is made up of 2 rings, which we can see one ring here and a second here, and that's the case for both adenine and guanine. Then we have cytosine, thymine if it's DNA, uracil if it's RNA. Those 3 nitrogenous bases are pyrimidines, which means that they only have 1 ring structure. So these bases have a complementary base, which means when you have the 2 DNA strands, the bases will align opposite a particular base, which is complementary to them. And thymine and adenine are complementary. Or if it's RNA, it would be uracil and adenine are complementary, so they'll always align opposite each other. And guanine and cytosine are complementary to each other, so they'll always align opposite. And what that means is you always have a purine and a pyrimidine base facing each other. And this complementary base pairing is really important because when the DNA replicates, it ensures that you get identical copies being made. Both DNA and RNA nucleotides, as I said earlier, can form a polymer, and the way that you go from having a monomer to having a polymer is a condensation reaction. And that is when water is removed, joining together 2 molecules, and a bond is formed. And the bond that forms is a phosphodiester bond. The polymer of the nucleotides is known as a polynucleotide, and that phosphodiester bond that forms is a really strong covalent bond. You end up getting what is known as a sugar phosphate backbone, which is where you have the sugar, which is here, the sugar, the phosphate, the sugar, the phosphate, the sugar, the phosphate, all attaching to each other, although that's actually showing ATP here, pointing out that you need energy for this reaction. So I'll do it on this one. We've got the sugar, the phosphate, the sugar, the phosphate. That's your sugar phosphate backbone, and it's all connected via these strong covalent bonds, the phosphodiester bonds. So you end up with a really strong polymer, which is important because you do not want that genetic code to be changed, such as mutations, because that is your hard copy of all of your genetic material. Next up, we have a look at ATP. This is a nucleotide derivative, so it's very similar in structure. The key difference is it contains 3 phosphate groups, so these 3 phosphate ions. You still have a sugar and a nitrogenous base, but it is always adenine and it is ribose. So you have adenine, ribose and 3 phosphate groups. And ATP is essential for metabolism. It is an immediate source of energy. When you break these bonds between the phosphate groups, it releases energy. And that can be used for all the different chemical reactions, which is what metabolism is, all the chemical reactions in the cells and also some processes. So the way this happens then is, first of all, ATP is made during respiration via condensation reaction, and it uses the enzyme ATP synthase. So we have ADP, which is diphosphate, which is when you have 2 phosphate groups, and you need to add on that inorganic phosphate. That's what PI is. So that inorganic phosphate group gets added on via a condensation reaction, and that's how you make ATP. And because it was a condensation reaction, water is removed. Now, to release the energy from ATP that's stored in those chemical bonds, you have to hydrolyze the ATP, and that is done using the enzyme ATP hydrolase. So this time it'd be a hydrolysis reaction, which is when you are bond to separate 2 molecules through the addition of water. So we're going to break the bond between the phosphate groups. That releases energy, and that then means we have ADP and PI. So breaking one of those bonds between the phosphate groups releases a small amount of energy. That means you don't have wastage energy. So if you need more energy, you would just break the bond between more ATP molecules. And that can then be used in chemical reactions. That inorganic phosphate group can also be added onto other molecules, and that's known as phosphorylation, when you add a phosphate group onto another molecule. And when you do that, it makes the new molecule more reactive and it gains energy. And that's actually what you see happening in the first stage of respiration in glycolysis. So if we have a look at DNA in a bit more detail, DNA, or deoxyribonucleic acid, that codes for the sequence of amino acids in the primary structure of a protein, which in turn is what determines that 3D structure and function of a protein, because it determines where the bonds can occur and therefore how it folds in that 3D shape. It's essential that the cells contain a copy of this genetic code, and it's really essential it gets passed on to new cells without being damaged, because if it does get damaged, you will change your DNA base sequence, therefore code for a different primary structure, potentially, and you end up with a non-functioning protein. DNA, as we said earlier, is a polymer, and the polymer is made up of 2 chains, which will then form a double helix. The nucleotide we talked about previously, you've got your phosphate group, deoxyribose, and one of those 4 nitrogenous bases. So the polynucleotides we also briefly talked about. So that is the polymer. Created by those condensation reactions, you get those phosphodiester bonds, which are the strong covalent bonds. The sugar phosphate backbone you can see a little bit clearer now because it's shown in blue around the outside of this double helix. And the DNA polymer occurs in pairs joined together by hydrogen bonds. So these coloured parts sticking out here are representing the complementary bases, and at every point that would be either 2 or 3 hydrogen bonds holding together those 2 chains. The hydrogen bonds can only form between the complementary base pairs, so you'll have those hydrogen bonds between guanine and cytosine and adenine and thymine. And as I said, you either have 2 or 3. Adenine and thymine can form 2 hydrogen bonds, whereas cytosine and guanine form 3 hydrogen bonds. So why are these complementary bases really important then? The 2 strands of the DNA molecules, first of all, are anti-parallel. And what we mean by that is they run in opposite directions. So on this side, you would have, this is the sugar phosphate backbone, so that would be your phosphate and the sugar at the bottom, whereas this side is upside down, you have the sugar and the phosphate on the top. You can't quite see it very clearly in this picture, but that is the concept of anti-parallel. And instead of saying top and bottom, you have it described as 5' and 3' end, which is referring to which carbon within the sugar ring is most exposed. So you'd have a 3' end and a 5' end, and then in anti-parallel it would be the opposite way round, so you'd have 3' and 5'. Now the complementary base pairing is important because it ensures the specificity of DNA replication and transcription processes. The 5 to 3' orientation of one strand will align with the 3 to 5 of the other one, and it allows the accurate copying and therefore the accurate expression of genetic information, which means that you have your correct DNA base sequence, which will then code for the correct amino acid sequence. Now, we talked about RNA as a nucleotide and as a polymer, and one of the polymers that RNA can form is messenger RNA, or mRNA for short. And mRNA is a copy of one gene. mRNA is made in the nucleus, so because it's a copy of one gene, it has to be copied in the nucleus because that is where the DNA is located. You get one copy of it, which we'll be looking at later on in this lesson in transcription, because that's how you make mRNA. It then leaves via the nuclear pore to the cytoplasm, where it will find a ribosome to attach to. So this is really important because DNA cannot leave the nucleus because it is so large, whereas mRNA is much shorter because it's only a copy of one gene. But this is actually a good thing. We don't want DNA to leave the nucleus because that is your hard copy of your genetic material. And if it got into the cytoplasm, there are enzymes in the cytoplasm that could break down that DNA. So instead, when the genetic code is required to make a protein, we make an mRNA copy of one gene. that can leave the nucleus because it's shorter. That can be used to create the protein. And if the mRNA breaks down, which it will do, that doesn't matter because you just make another copy of it from your DNA. So it is short-lived because it is going from the nucleus into the cytoplasm, where it will get broken down after it's been used in protein synthesis. And it is single-stranded. It forms a straight line, and every 3 bases on your mRNA sequence is known as a codon. So 3 bases, 3 consecutive bases on mRNA, which codes for one amino acid, a specific amino acid, is known as a codon. Next, we're going to have a look at how DNA replicates, and it replicates through semi-conservative replication. And what we mean by that is each time the DNA replicates, one strand is conserved and one strand is newly synthesized. So it's semi-conservative, but it's really important that you say one strand, not half, because half could imply it all breaks apart and mixes up and half is new, half is old. So you have to specify one strand is new, one strand is old or conserved. It's in this stage that mutations could occur because sometimes copying errors do happen in DNA replication, and they are random, spontaneous, and that would result in a change to the DNA base sequence and therefore would code for a different amino acid potentially. But we're going to come on to that right at the end of the video. So this happens in S phase of interphase of the cell cycle. And this concept of the 3' and 5' end are useful in describing the stages of DNA replication, because instead of saying the start or the end, you can talk about the 3' or 5' end. And this is relevant because we're going to look at the enzymes involved in DNA replication, and the enzyme that catalyzes one of the stages of DNA replication is complementary in shape, so the active site is complementary in shape to the shape of DNA only at the 3' end. And that means that that enzyme can only attach at the 3' end of the DNA. So describing the stages of DNA replication could be a potential long answer question. There's 4 key steps. Step 1 is the DNA, first of all, has to be separated apart from that double-stranded double helix. And that is done by the enzyme DNA helicase. DNA helicase breaks the hydrogen bonds between the complementary base pairs, and that is what causes the double strands to unzip, unwind, and you get these replication forks, which is what we can see here. It's actually labelled slightly differently on the diagram, but there's our DNA helicase breaking those hydrogen bonds, and you get one strand over this way, one strand over that, and that is known as a replication fork. Step 2 then, those 2 separated DNA strands will now both act as a template for new DNA strands in DNA replication. Free-floating DNA nucleotides that are always within the nucleus will align opposite their complementary bases, and they'll form hydrogen bonds between those complementary bases. But at this point, the nucleotides aren't joined together with that phosphodiester bond. And that is where the enzyme DNA polymerase comes in. DNA polymerase is able to join together those adjacent DNA nucleotides through the catalyzation of the condensation reaction, and it forms the phosphodiester bonds. And that then results in 2 daughter DNA molecules, each consisting of one of the original parental strands and one newly synthesized strand. So one key focus is knowing the process of DNA replication, but you also need to know the roles of different enzymes involved. And we've talked about DNA polymerase. We said that DNA polymerase catalyzes the formation of the phosphodiester bond between those adjacent nucleotides on the newly synthesized chain. And this is the enzyme where I was saying it is complementary in shape to the 3' end. So DNA polymerase will always bind to the original template strand at the 3' end. That means the newly synthesized strand, which is anti-parallel, will have new nucleotides being added in the 5' to 3' direction. And that leads to differences between the leading strand and the lagging strand in DNA replication, which if I just go back to this diagram here, we can start to see that concept of leading strand versus lagging strand. So the leading strand is where you do have this continuous addition of the bases being catalyzed with that condensation reaction. The lagging strand, you get it in separated sections, which we call Okazaki fragments. And that's where we see the role of DNA ligase. So DNA ligase is involved on the lagging strand during DNA replication. So DNA polymerase synthesizes short DNA fragments called Okazaki fragments in that opposite direction of the replication fork movement. And as a result, those sections, those Okazaki fragments, have to be joined together. And that is what DNA ligase does. It joins those Okazaki fragments by catalyzing the formation of phosphodiester bonds, completing the synthesis of that lagging strand on the DNA molecule. Next, then, we have a look at protein synthesis. So for protein synthesis, the first concept is you need to have an understanding of the genetic code. And there's three special features. The genetic code is degenerate, it's universal, and it's non-overlapping. What we mean by degenerate is amino acids. There are 20 amino acids, but there's actually 64 triplets of bases. So amino acids can be coded for by more than one triplet of bases. Universal is that the same triplet of bases codes for the same amino acid in all organisms. And lastly, non-overlapping. This is when each base in a gene is only a part of one triplet of bases, or you could describe it as a discrete unit. So each codon is its own separate discrete unit. There's no overlapping of the bases in multiple triplets. So the advantage of this is the fact that the genetic code is degenerate means even if a mutation occurred, changing one of the bases in a triplet, that new triplet due to the mutation might still code for the same amino acid. So it reduces the impact that mutations might have. The fact that it's universal has been really helpful in genetic engineering because it means that we can take a human gene and insert it into the DNA of bacteria, and that bacteria will then produce the human protein. Non-overlapping is also linked to the concept of mutations. So it's really useful that each base is only part of one triplet, because if a mutation did occur in a triplet, then at least it's only one codon which is affected and therefore only one amino acid. So protein synthesis occurs on the ribosomes or the rough endoplasmic reticulum because they're rough because they have ribosomes. Transcription actually happens in the nucleus, and that is the first stage, and that is where the DNA is copied into an mRNA sequence, so that's just for one gene. Translation is the part that happens on the ribosome, and this is where the polypeptide chain is created. So we're going to go through those two processes, but just a few other key things that you need to know. Number one is what introns and exons are. So introns are sequences of bases on the DNA, so in a gene in particular, that do not code for amino acids and therefore the polypeptide chain. So these introns have to be cut out or removed, or spliced is a technical term, out of the mRNA once the copy of the gene has been made. So mRNA is made and then the introns are spliced out. That then just leaves the exons behind, and exons are sequences of bases in the gene that code for sequences of amino acids. So that's the coding section that's needed to code for the polypeptide chain. There's also start and stop codons. So at the start of every gene, there is a start codon, which is 3 bases, which will code for a particular amino acid, which will initiate translation. The stop codon is the final 3 bases at the end of every gene, and these will not actually code for any amino acid, and as a result it causes the ribosome to detach and therefore it stops or ends translation. So let's go through the 2 stages then. Transcription is the first step, and this is the process that's happening within the nucleus where we get an mRNA copy of one gene on the DNA being created. So the steps are very, very similar to DNA replication. First of all, DNA helicase breaks the hydrogen bonds between the bases in the 2 DNA strands to unwind that double helix and to separate the 2 strands. Here's the first difference though. Only one of those strands will act as a template, whereas in DNA replication, both of those strands of DNA will act as a template. Next then we have 3 mRNA nucleotides lining opposite the exposed complementary bases on the DNA. Same concept, but this time it's mRNA nucleotides instead of DNA. And then the next bit, again, is very similar. It's just a slightly different enzyme. This time it's RNA polymerase that will join together the adjacent nucleotides, forming a phosphodiester bond through that condensation reaction. And that is how you then get an mRNA polymer. And once that one gene is copied, then you would have the introns spliced out. And then the mRNA can leave the nucleus via the nuclear pores and go and attach to a ribosome in the cytoplasm. And that then takes us on to the next step, which is translation. So once that modified mRNA has left the nucleus via the nuclear pore, it attaches to the small subunit of the ribosome at the start codon. tRNA molecules, which are a different type of mRNA that carry specific amino acids, they have anticodons on them, which are 3 bases complementary to a codon on mRNA. So the tRNA molecule with the complementary anticodon to a codon will align opposites, and the ribosome is able to hold 2 tRNA molecules at a time. The 2 amino acids that will be at the top of the tRNA molecule are therefore delivered and is specific based on the anticodon and the codon, and they are then able to join via a peptide bond, and this requires an enzyme. And it also requires ATP, energy released from ATP, to form that peptide bond. The tRNA is then released and the ribosome will move along the mRNA molecule to the next codon, and that process happens again. So the next tRNA molecule can then align opposite its complementary codon compared to its anticodon, bringing a specific amino acid. And this process continues until the ribosome reaches the stop codon at the end of the mRNA molecule, and that is what causes the ribosome to detach and therefore ends translation. That then results in your finished polypeptide chain, which has been created, which can then enter the Golgi body for further folding or modification, such as maybe adding a carbohydrate to it. So the last concept in this module is gene mutations. And a gene mutation is a change in the base sequence of DNA that might result in a different polypeptide. They occur randomly and spontaneously during S phase of interphase, which is when DNA replication happens. But these mutations are more likely to occur if you're exposed to certain chemicals or high energy radiation. So for example, UV light, gamma rays, X-rays, and the chemicals we call carcinogens, so mustard gas, for example. A gene mutation could result in either a base being deleted or substituted. So for example, we've got our original DNA base sequence here, and we can see that base cytosine has been substituted, so changed, for the base adenine. So that is a substitution gene mutation. The next one we've got is a base has been added or inserted. It's exactly the same sequence except thymine has been added at this point. So that means we've now changed all of the subsequent triplets after that mutation, and we've also got an extra base just not a part of any triplet. We call that a frame shift when it shifts along like that. Deletion has a similar effect. If you delete one of the bases, all of the triplets after the point of the mutation change, and that means it's a frame shift. This time, though, it's all of the triplets move back one position. So this one, we don't even have a triplet anymore, so that wouldn't even code for an amino acid. So you're coding for different amino acids, and you'd have one fewer. So sometimes a substitution can be described as silent, meaning that the new codon still codes for the same amino acid. And that's because of that feature of the genetic code we talked about, it being degenerate, meaning that multiple triplets of bases can still code for the same amino acid. However, those deletions or insertions, those can result in frame shifts where after the point of mutation, all of the subsequent codons are changed and therefore will probably code for different amino acids, and you'll get a very different primary structure or sequence of amino acids. Therefore, the protein will fold into a very different shape and it will be non-functioning, or it might have a completely different function. And in terms of enzymes, that would mean the active site would be completely changed and the enzyme would no longer catalyse the reaction it's meant to. So that is far more harmful. So that takes us to the end of topic 6, going through nucleotides, DNA, RNA, DNA replication, ATP, protein synthesis and DNA replication. Hope you found it helpful. Don't forget to subscribe so you don't miss out on any of the latest videos, in particular all of these entire topic videos to help.